Now, he is, Aguzzi is one of the giants in the prion field. Just to go over sort of a few of his most recent achievements, he invented the first in vivo model of microglia depletion and was thereby able to sort of help establish that microglia, they possess a, anti-prion activity. And he will be going a little bit into this today by looking at expression of a PrP in different cell types and how that influences pathology. Now, he also he identified the cell types and molecules that enable invasion of prions into the brain, and this was done in a series of articles in Nature, Neuron, Nature Medicine, Science, and so on, starting over 20 years ago. And this included the discovery that follicular dendritic cells are so-called prion factories. So these are cells that are sort of tightly associated with the gut and are very important for sort of, enhancing replication of prions early on in the infection process. Now, he identified also plasminogen as a prion carrier in blood, and has also improved the process of differentiating prion strains by using fluorescent molecules, and in particular polythiophene, which he identified some years ago that had very prominent anti-prion activity. And he has also seen recently that they also can prevent a beta amyloidosis. So these are sort of beta seed binders that look very promising. In some ways, unfortunately, that there's no IP on these compounds, so industry is not particularly interested in taking this into the clinic, but hopefully there will be some government funded approaches that will test this in more detail in the clinic. Now, also, for those of you who are interested in Schwann cells, he also found that PrP makes, basically PrP that is made by neurons can prevent Schwann cells from developing demyelination. So this is sort of has therapeutic implications also for MS and other diseases. And, I just want to keep this short. He has published over 400 scientific articles. So I think that New York would actually be a very nice city for him to live in, the scientist that never sleeps, in the city that never sleeps. And with a reference over 47,000 times, and, AIDS index, well above 100 and so on. So, one of the most cited, he is actually the most cited neuropathologist in German-speaking countries and the third of all neuroscientists. And we all know the quality of science that comes from German-speaking countries. So this speaks also very highly about his scientific acumen. Aguzzi, thank you very much for taking the time of your busy schedule, and we look forward to your talk.
Thank you. Thank you, Einar. Let me see. Can you guys hear me? Yeah.
Yeah.
Perfect. So great to be here, and no, it's a pleasure and an honor, and I see a lot of old friends here, and it's great to connect, if only virtually. Although I have to tell you one thing that I think we all discovered in the last 1 year or so, that we can have incredibly intense and frequent interactions across the world with these video conferences is something that we never did before, but it's it's not the same as personal interaction, but it's certainly better than non-interacting, and so it's something that I am enjoying tremendously, and I really want also to thank you all for coming to my seminar, and I very much hope that this will be as interactive as possible. So don't hesitate to stop me in the tracks and to ask questions, and that's the whole point. And so So I want to tell you a couple of stories of what my lab is doing these days, and essentially these are usually people say, yeah, I'm going to talk about unpublished data. These are pre-published data, so to speak, because I'm going to show 3 stories that are all on bioRxiv papers that have been put on the web a few weeks ago, and all 3 actually since the beginning of the year 2021. So the first story relates to something that is really neuropathology, and the reason why I embarked in this project together with an MD PhD student, Daniel Kirschenbaum, who has recently graduated and has moved now to the lab of Ido Amit at the Weizmann Institute in Israel, and Francesca Cappo is an Italian PhD student. And the reason why we embarked into this is the following. When a neuropathologist looks at a histology slide, he actually sees a 2-dimensional image, but what the pathologist is doing in his brain is to try and reconstruct the 3-dimensional makeup of the tissue that he's examining. And that works actually remarkably well if you think how hard this actually is, but it's obviously, it has a lot of limitations. And so I became very interested in trying to analyze brains holistically. And in the case of amyloidosis of the brains, be it prion diseases or Alzheimer's disease, one idea was, okay, would it be possible to enumerate every single plaque, every single amyloid deposit that exists, for example, in a mouse brain, and if we were able to do this in a natively digital format, maybe we can actually use a pattern recognition system derived from machine learning and artificial intelligence that could allow us to see things that we were not aware of before. So that was really the goal. And as you know, there is this famous technology that was pioneered by Karl Diceroth in Stanford that is called Clarity. And essentially, the way Clarity works is that in this case, a mouse brain is put into a buffer, and then by electrophoretically, the lipids are being removed from the brain and substituted with a hydrogel. And by that, the brain becomes transparent, because the real reason why a brain or why any tissue is not transparent is not the proteins. The proteins, a protein solution is opalescent, but it's essentially transparent. The problem is that the moment you have an interface between a lipid and water, you will have a refractor, a change in refraction, and that scatters the light, and that is why you can't see through the tissue. So by pulling out the lipids, you should actually be able to make the tissue transparent. But one thing that was found over the years is that this technology doesn't work very well. And Daniel Kirschenbaum had this intuition that the reason why it doesn't work very well is maybe because the dielectric constant, the resistance, the electrical resistance of the tissue is much higher than that of the surrounding buffer. So the idea was, okay, if you were, and this is really what he did, and it sounds a bit trivial, but actually it really changed our game completely. He separated the anodic from the cathodic buffer with an insulator, and now you have two separate circuits here, and the stream of electrons now will have to pass through the tissue because it has no other way to go around it, and this enormously increased the the clearing potential. And in fact, now we can make a brain, mouse brains can be made transparent within a few hours, and small human biopsies can be made transparent within minutes, which is something that could even enable clarification technology as an intraoperative technique. So, essentially, we have this problem was more or less resolved, but then the next problem is how do you stain all these tissues? And there again, Daniel and Francesca Catto have done a lot of work, and they came up with an idea of confining a dye to a low melting agarose block that is essentially cast in front of the brain, and again use electrophoresis to drive the dye into the tissue. That again works now in the meantime very, very well. And what kind of dyes do we use? We use something that is called, we call it luminescent conjugated polymers. And these compounds were made by Peter Nilsson, who used to be a postdoc in my lab some 10 years ago. He's an organic chemist. He's now a professor at the University of Linköping. And Peter had this brilliant idea of using these compounds that you can see here, which to stain amyloid. Now, these compounds are highly fluorescent because they contain a conjugated electron system, which means that you have double bond, single bond, double bond, single bond alternating, and this creates a pi electron cloud, which is the basis for the fluorescence. And but the interesting thing about it is that the connection between the thiophins is a single bond, meaning that these molecules have rotational freedom, and then, for example, if you put in residues, for example, carboxylic residues here, the ionization state will determine the geometry of the polythiophene. So if the polythiophene repel, you will have a planar backbone. If they attract, you will end up with an orthogonal backbone. And the amazing thing is that the emission spectrum of the fluorescence is dependent on the geometry of the thiophens. And in fact, this is always the same. This is polythiophenic acetic acid at 3 different pHs. Of course, the protonation state of the carboxylic residues changes, and the result is that you end up with different colors. Now, when Peter came to my lab many years ago, his idea was, well, the distance between the thiophene rings is around 5 angstroms, so maybe they might actually intercalate with the amyloid, which stacks at around 10 to 11 angstroms. And this turned out to be indeed the case. And in the meantime, we have also, together with Beat Meyer, we have recovered the high resolution solid state NMR structures. And this, for example, shows you how a polythiophene interacts with an amyloid. This is yeast het S prion, and what you see is that there is a hydrophobic groove here in which the thiophene rings enter the groove. So the thiophene is oriented axially to the fibril, and then here this carboxylic residues, which are negatively charged, interact with lysines on the surface of the fibril, and this gives rise to an incredibly strong interaction, because well, each of these interactions are very weak, but they are cooperative. And then we also used in silico docking to generate further generation of polythiophens that would bind particularly well. And then so these are the substances which, first of all, they are low molecular weight, and secondly, they are very highly charged. So we thought maybe this is exactly what you should use to stain plaques. And Daniel has created a little machine that can be 3D printed, and it's very simple. Essentially, you put in the tissue, and then you use this cast agarose to push the the dye into the brain using electricity. And then the next question is, how do you record the images from these tissues? And there we decided to use selective plane illumination microscopy, which is also called light sheet microscopy. And essentially the trick here is that the light comes at a perpendicular angle to the acquisition path. So you have 90 degrees between the light and the objective, and essentially what the light does, it through a combination of mirrors and lenses, it creates a sheet, an undulating sheet, which will cut an optical section through the tissue, and then now the tissue is being moved, and this way it's a bit like a CT scan or an MRI scan. You can acquire the entire tissue at a resolution which in our case is around 3 microns isotropic, so XYZ 3 micron resolution. And this is the machine. I know it doesn't look very much like a microscope, but that's what it is. And then nothing like this existed commercially. So together with Fabian Vogt and Fritjof Helmchen at the Institute of Brain Research in Zurich, we actually made this machine. And here you can, in this paper, it's actually very nicely written how to make it. There is also a shopping list. The most expensive part are the lasers. Here you see actually the guys have put all the different, or some labels so you know where is what. Here is actually the camera, and here are actually the lasers, the fiber optics that comes in there. Here is the chair, in case you don't know about it, but I mean that you guys try to make some fun about this. But then we bought a lot of different lasers. As you see, we like the many colors, and actually they also ran a time-lapse video of them. It took them 2 days to put together the microscopes once we received all the materials. So if you want to do this at home, you are very welcome to use this instructional video to assemble a light sheet microscope. And then once all of this done, then you can actually look at the plaques. And in this case, this is an APP-PS1 mouse. It's a transgenic mouse model for Alzheimer's disease. And what you see here are all the yellow guys are actually plaques. The red stuff is Claudin 5 GFP transgene made by Christer Betzold at Karolinska, and so that we can actually image all the brain endothelial cells. So, and the idea was really to inspect let's say the connection or the relationships between the vessels and the plaques. And here, after segmentation, you can actually see all the vascularization of the brain and all the plaques. And now, having this kind of data sets allows you to do a lot of things. Like, for example, asking the question, what is the average distance of each plaque to the nearest vessel? And this was important because, for example, small molecules like beta secretase inhibitors so are very likely to diffuse into the brain relatively easily, but large molecules like antibodies won't do so. And therefore, the diffusion distance from capillaries to plaques may be important for therapeutics. And this led us to formulate the question whether current therapeutic options for Alzheimer's may actually exert different effects in different sites of the brain. And in order to examine this question, we looked at 3 different therapeutic modalities. We use BACE inhibitors, we use antibodies, And we use also the polythiophins, which we had previously shown to be very effective against prions. So mice were treated for 3 months with each or either of these 3 therapies. Then the brains were clarified in the way that I have explained to you. Then they were registered to the Allen Brain Atlas, so that we would have a standardized ways of looking at each single voxels. And then we asked, okay, how effective was the therapy? And this is the results. This is so first of all, what we do is we compare the distribution of plaques in placebo-treated mice to mice treated with whichever, with one of these 3 compounds. And from this, we derive a probability, a 3-dimensional probability map. Now, what you see here, you have to be careful. It's very abstract. What you are seeing here are single voxels, and each data point, each is colored, and the color map corresponds to the efficacy of treatment. So so we are not looking at plaques here. We are looking at the probability that in a particular voxel the treatment has been effective. And this is a summary from 10 mice treated versus 10 control mice. Then we can do the same thing for a different treatment. So we can look at BACE inhibitors and, for example, polythiophins, and then we can ask, okay, what is the difference? And this is now the combined map that tells us that the treatment may have different relative efficacy in different areas of the brain. And this is then you can do lots of transformations. For example, you can make optical sections and then actually look at where in the brain the treatment has been effective. So here we are looking actually at the areas where the brain has been cleared of plaques. And the results, I have to say, that I found this totally mind-boggling. For example, here we are comparing inhibition of beta secretase to treatment with a polythiophene. And what we see is that both treatments are very effective, but the reasons in which the treatments are effective are completely different from each other. For the BACE inhibitors are almost exclusively effective in the basal portion of the cortex. The dorsolateral cortex is very, very poorly cleared, and the polythiophene do exactly the opposite. Here you have a lot of clearance, and here you have very little therapeutic effect. Now, for me, this is really exciting, and it points to something that I think was not really appreciated so far, and that is that different therapeutic options are very profoundly, have very profound differences in their in the topology of their effect. Now, I have to say that this analysis is by no means trivial, and what you have seen thus far is just simply these 2 images are the summary of more than 100 terabytes of data, and this really stretches also the computational part to the real limit. And in fact, we have been analyzing this data together with a group of fantastic computational scientists headed by Jin-Hyung Lee in Stanford. And it turned out that it was impossible to push the data between Stanford and Zurich via the internet. So we resorted to actually writing the data on hard disk and sending the hard disk to Stanford by FedEx. And right, so there is some background noise. Maybe
if we can have everyone mute themselves while Professor Aguzzi is speaking, that would be great. Thank you.
Okay, the Zoom has translated my name as Professor Goofy. All right. So then I had this idea that maybe we can. Maybe we can use this topological information on plaque clearance to discover something about the genetics of Alzheimer's pharmacology. And to do that, we consulted several transcriptomic atlases, so about spatial transcriptomics. And the idea was to correlate the loco-regional efficacy of Alzheimer's treatments to the expression patterns of the entire transcriptome in the brain. And this analysis was done for each single gene expressed in the brain. And this was done mostly by Ehsan Dalgani, who is a PhD student with Jean in Stanford. And the results are very fascinating. So this is essentially this distribution. This MI index shows the amount of similarities between each gene and the clearance of plaques. And what you see is that in the case of the BACE inhibitor, the one gene that has a very strong similarity is the expression of Thy1. So why is that? Well, the reason is trivial, but it's a great internal control, because the APPPS1 transgene that is driven by the Thy1 promoter in this mouse model. So you would expect that the clearance of plaques can only occur where the plaques are there. And this is, of course, only in the regions where you have Thy1 expression. So it's not surprising that Thy1 expression correlates very well with plaque clearance. Another gene that is very strongly and very nicely expressed is BACE. BACE itself, well, NB360 is a BACE inhibitor, so you would expect that it is mostly effective in those areas of the brain where BACE is most highly expressed. So that is another nice control. And then you have other genes, and CX3, CL1 is one of them, and then there is a bunch of others. And here there is an entire map. What we have done here is to cross-correlate the expression, the similarity maps of the transcriptome to BACE inhibition to the similarity maps of the transcriptome to inhibition by a polythiophene. And you see that actually the genes are very different. In the case of the BACE inhibitors, you see a bunch of myelin gene coming out, myelin basic protein, protolipid protein. And in the case of the polythiophenes, you see neuronal genes that are coming up. So that is, this is where we stand now. And I believe that there is a lot of value that can be gained by this technology. And in the next step, we are moving to humans. And this is now a human hippocampus, and it was treated in exactly the same way. So it's an autopsy material, and this is the work of Anna Maria Royce, who's an MD PhD student in my lab, and she has found a way to, or she had tweaked the protocol in such a way to actually clarify human tissues very effectively. And here she has stained vessels, capillaries, arteries, and A-beta plaques, and in 3 different colors. And this is a patient suffering from Alzheimer's disease. And this is again a 3D light sheet reconstruction of a hippocampus with from a human suffering from Alzheimer's disease. And of course, and then you can do much more, no? Because once you have this data set, you can actually segment it and identify the vessels with a very high degree of realism, And once you have all this entire data set, then you can do blood flow simulation. And this is something that we are doing together with the machine learning department at the ETH in Zurich. And Petros Komoutsakos is the head of machine learning and chair of machine learning in Zurich. And he has become very excited about this. He is one of the best, or the world's best persons in flow simulations, and the hope is that we can actually model the hemodynamics of in human brains affected by Alzheimer's. Right, the next question that, or the next chapter that I want to talk about relates to the molecular basis of prion toxicity. And this is mostly the work of Carl Fronzek and Ashwin Lakkaraju, and I will show you something about it. So really, as Einar has mentioned, in the last several years, we have been focusing on the normal function of the prion protein, and we found that it is important for myelin homeostasis, and specifically we found that the prion protein interacts with there is some background noise, so if you, yeah, perfect, very good. And so we found that the N terminus of the prion protein interacts with a G protein coupled receptor called GPR126. But then this really did not answer the question of why the prion protein is toxic. And now I have to go back to an experiment that Sebastian Branner did in my lab 25 years ago. Sebastian was my first postdoc. He is now active in London, and what Sebastian did at the time was to graft a piece of PrP overexpressing brain into the brain of a PrP knockout mouse. And then when he gave prions, he saw that there is a spongiform encephalopathy that arises in the graft. And you see here very nicely all these holes. It looks like a sponge, so that is why it's called spongiform. But although the prions would diffuse everywhere into the brain, the pathology was restricted to the graft. And at that time, we thought that this might indicate that the pathological prion, the aggregates, the PrPC, may require PrPC in order to damage neurons. And so that was one line of research. And then another thing that Frank Heppner did also exactly 20 years ago is we became very interested in antibodies, and I thought that maybe we could stop prion diseases with antibodies against PrP. And in fact, this work that Heppner did was actually even before the whole frenzy about anti-A beta antibodies. It was really one of the papers that kickstarted the field. And in fact, Heppner showed that it is possible to prevent the transmission of prions from the periphery to the body, to the brain, using a recombinant antibody. And in this case, the antibody was genetically engineered. However, something that we had to realize was that some antibodies were protected, but other antibodies were super neurotoxic. And this is one such antibody. This one such antibody is called POM1. And here, what we have done was to culture brain slices, cerebellar organotypic slice cultures on a support membrane. And what this is a transversal section, and you see here the cerebellar granule cells, the molecular layer, the Purkinje cells, and you can keep these slices alive for several months, and they survive very, very nicely. But if you give this antibody, within 3 days, the slice is completely sintered, and the neurons are gone. And this happens even if you create a single chain FV version of the antibody, will also be ultra toxic. And then we found that this antibody binds to a region of around 400 square angstroms on the surface of the prion protein. This is the prion protein. This is actually the FAB1 version of the antibody with the heavy and the light chains, and this is really this is the binding site. And this is not a model; it's a real crystal structure of the complex of the antibody with the prion protein. And my hope was that from that we would be able to understand really how toxicity comes about, but we didn't really get there. And then I met Luca Varani, who does molecular modeling in Bellinzona, and Luca was telling me, the crystal structure doesn't tell you the whole story. You have to do molecular dynamics because the crystal tells you it's just a snapshot of what is going on. And so Luca made many simulations, and what he found is that this floppy loop, this unstructured loop of the prion protein, does not really interact with this alpha helix here because the distances are too long. But when the toxic antibody comes in, then what happens is that this arginine is shifted in such a way that it will interact with this histidine 140 on the loop, and therefore this loop becomes extremely rigid. You don't see this in the crystal structure, but in the molecular dynamics, this becomes very obvious. And because this becomes very rigid, then other parts of the molecule become there is a long range effect, and the other parts of the molecules become more floppy. And the question arose, well, maybe this is the reason for toxicity. Maybe here there is a subtle molecular change that drives toxicity. I should also say that the toxicity of this antibody reproduces the toxicity of prions. So we call this POM1 antibody a prion mimetic, and there are many reasons. I won't go into the details, but we are absolutely convinced that we are reproducing the same phenomenon that happens in prion infections. Now, how to test this idea that this salt bridge that occurs here, it may really be the cause of toxicity? Well, I thought that maybe we can mutagenize the POM1 antibody in such a way that we can, there is a network of salt bridges here, and we could mutagenize every single antibody, every single residue in the antibody that interacts with the prion protein, and make a version of the POM1 antibody that will still bind, but is unable to fix this conformation. And this is what was done here. So Karl Fronzek in my lab has mutagenized both the heavy chain and the light chain. And these are all the different mutants. And then he has measured the affinity of the mutants for the prion protein, either by FRET or by surface plasmon resonance. And this is the affinity of the wild type. And when you mutagenize the antibody, you lose some affinity, but not much. I mean, some of the mutants have a lousy affinity. I mean, these are essentially useless. But some of them actually have mostly retained the affinity, and yet they cannot induce this latch. We call it the hydrogen latch here, and the results are amazing. So here is what happens. This is a organotypic cerebellar slice culture exposed to non-infectious brain homogenates. And here you see very nicely that the structure of the cerebellum is still fine. If you infect the prions with Rocky Mountain Laboratory prions, then the slice becomes damaged. You see that the arbor vitae, the granule cell layer of the slice is demolished. Now, if you now here we're looking at several of our of our antibodies, and it turns out that this antibody has quite a good affinity, and you see that it protects from the prion infection, and this is amazing. This is really the tyrosine 104A in the heavy chain, the mutation. This retains the same affinity as the native antibody, and it completely protects the slice from neurodegeneration. So for me, this is really remarkable, because essentially what Carl has done was to take a super toxic antibody, make a single residue substitution, and completely revert the polarity, and going from a toxic antibody to a protective antibody. Now, why is this protective? Now, my thinking is that this interaction mimics what happens when the pathological prion, the misfolded protein, encounters the normal prion protein. So my thinking is that the antibody POM1, the native POM1, will induce this change that is neurotoxic, just like the prions also do. Now, when you mutagenize the antibody, what it does is that you now have essentially a competitive antagonist. So an antibody will enter this pouch, will enter this pocket, and will prevent the pathological prions from occupying the pocket, and that is why it might be protective. All right. There's a final story that I want to share with you as well, and this is vacuolation. And I am these days I'm super excited about vacuolation, but I have to say that I have been wondering about spongiosis for 30 years. I mean, the first time I saw the histology of a patient with Cerebellar disease, I was amazed. I mean, the entire brain is ridden with these little vacuoles, and this is very specific. It's actually, if you see this in histology, you know within a fraction of a second that you're dealing with a prion disease, because this is really how prions look like under the microscope. So the question for me during all these years has been, what is it that causes vacuolation? What is the molecular pathway that damages the brain to the extent that you have all these vacuoles? And so first of all, the vacuoles are inside the neurons. You see here, there is a myelinated neuronal process, and then all of a sudden the process expands, and then you see these vacuoles, you see some multilamellar organelles that are degenerated inside the vacuoles, but the important thing is the vacuoles are inside the neurons. So what are these vacuoles, and how do they come about? Well, that was the question that I asked to my postdoc Ashwin Lakkaraju when he joined my lab some 6 years ago, and so I told him, look, Ashwin, why don't you go and find out why the vacuoles come about? And I can tell you that Ashwin, who is an amazing cell biologist, really an incredibly thoughtful and innovative person, he really went up, and I would argue that he has resolved the issue. And what I'm going to say to show you, in my opinion, it may well be the most important thing that my lab has done in the last 30 years. But I have to say that I probably stand pretty alone with this judgment, because we are having a hell of a lot of trouble publishing this data. But I tell you that I'm very convinced that this is very important. So essentially, what Ashwin has his intuition was that these vacuoles may have something to do with late endosomes and lysosomes. And there is a complex of proteins with phosphoinositol kinase called PIKfyve and a phosphatase called PIK4. And what PIKfyve does is that it takes phosphoinositol 3-phosphate and converts it into phosphoinositol 3,5-diphosphate. And all of this happens on the outer surface, on the cytosolic surface of endosomes. And the size of the endosomes is controlled by the concentration of PI3-5 diphosphate. And in fact, if you knock down PIKfyve, you enlarge the endosomes. And very recently, just a couple of months ago, a structure, a cryo-EM structure of PIKfyve, of the complex of PIKfyve, was published. It's a beautiful structure. You see that what PIKfyve does is that so there is VAC14. VAC14 is a scaffolding protein. It makes this beautiful star-shaped pentamer, and then PIKfyve sits on top of it, and FIK4 is a phosphatase, and FIK4 dephosphorylates PIKfyve and essentially activates it. Now, this is what happens if you do a shRNA experiment. This is a LAMP1 stain, which is a late endosome lysosome marker. And you see here all the little lysosome in normal cells. But if you knock down PIK5, you see that the lysosome starts growing. They make essentially backwards. So Aswin's idea was, could it be that PIK5 and its catabolites may play a role in the vacuolation, in the spongiosis of Creutzfeldt-Jakob disease? And then he did an experiment, which I tell you, it literally wiped my socks off. I mean, I couldn't, I really had a hard time believing it, but look at this. These are mice that have been infected either with non-infectious brain homogenate, so this is the control, or with prions. So this is the Rocky Mountain Laboratory Passage 6 prions. And look at what happens to PIK5. You infect the mice with prions, and PIK5 disappears. And it's selective, because I told you PIK5 exists as a trimeric complex with, or a ternary complex with FIK4 and VAC14. FIK4 is a phosphatase, VAC14 is a scaffold. And what you see is that FIK4 is still there. VAC14 is still there. It's only PIK5 that disappears. And the same happens also in humans. This is human brain tissue of victims of sporadic Creutzfeldt-Jakob disease. Again, you see PIK5 here in the controls, and you see that PIK5 disappears in the brains of CJD victims. And the interesting thing is that it mostly, you see that there is much more depletion of PIK5 in type 2 Creutzfeldt-Jakob disease, which is the most severe form, and whereas in type 1 there is a bit less. I mean, the disappearance of PIK5 is less pronounced, and type 2 is really the one with a very severe spongiosis. So this is preliminary evidence, but it's very exciting. And so in order to understand really the molecular biology of it, you need a cellular model. You know, you cannot do this in a mouse. And for that, we used a cell model called GT1-7 cells. And these cells, they create microvacuolation upon prion infection. So here is, again, the cells are infected with non-infectious brain homogenate, not much happens. But if you infect them with actual prions, you see all these vacuoles that are now coming up in the cytosol of the cells. And then the question is, what happens to PIK5? And this is beautiful. Again, non-infectious brain homogenate, loss of PIK5. You infect the cells with RML prions, PIK5 goes down. Now, this is really interesting. And then the next question is, we have this model with cerebellar organotypic brain slices, and again we wanted to see what happens. And here we have given POM1. POM1 is the toxic antibody that I showed you before. Look at what happens. IgG is the control, pooled immunoglobulins, PIK5, you give POM1, PIK5 disappears. And this is actually the quantitation of this plot. And again, it's totally specific. FIK4, VAC14, and actin are not affected. So, which again reinforces my contention that I explained earlier, that POM1 is a prion mimetic antibody. It creates the same pathology that is given by prions. So it's very specific. POM1 is continuously, I mean, whatever you do, you infect with prion, you give POM1. In all cases, you push down PIKfyve. Now, how does this work? What is the pathway that starts with prion infection and leads to PIKfyve removal and to vacuolation? Well, this is the work of Giovanna Mallucci, who showed that in prion infections there is a fair amount of ER stress that activates the PERK pathway of phosphorylation of EIF to alpha and inhibition of protein synthesis. So we were wondering whether ER stress may be actually upstream of this. And indeed, in this experiment, Aswin has shown, and I won't go into much detail, but the bottom line from all this is that the ER stress, at least in a temporal manner, precedes the loss of PIK5 protein. And this may indicate that ER stress is really what drives the disappearance of PIK5. And indeed, what he showed is that if you give this JSK compound, which is an inhibitor of the PERK pathway of ER stress, you can actually block, at least to some extent, the disappearance of PIK5. You see, this is normal level of PIK5. You infect with prions, PIK5 disappears. But if you infect with prions and deliver GSK, then you see that the amount of PIK5 are at least partly rescued, and also the amount of vacuolation is diminished. Right. So what happens? Aswin's idea was, why does it disappear? And well, one possibility is either less PIK5 is made or the degradation of PIK5 is enhanced. And what was done here was to induce ER stress using subsigargin and measure the half-life of PIK5 using metabolic labeling in a pulse chase experiment. And what turns out to be the case is that in the presence of ER stress, the half-life of PIK5 is reduced in a major way, and PIK5 becomes hyper-ubiquitinated. So the interpretation of this is that PIK5 is destabilized by ER stress. But how does this work? What does it mean that is destabilized? Well, and here again, I have really to commend Aswin Lakkaraju, because he had this wonderful idea. He thought, well, you know, I mean, many proteins are, their stability is controlled by acylation. So could it be that PIK5 is acylated? It would make sense, because phosphoinositol is inserted into the membrane. PIK5 is localized at the surface of endosome, so it maybe is anchored to the membrane with a palmitoyl residue. So he tested this using a methodology that is called Acyl-RAC. I will not go into the detail, but the bottom line is indeed, it turns out that PIK5 is an acylated protein. So so how do proteins get acylated? Well, there are 23 enzymes called DHHCs. These are the acyl transferases in the mammalian genome. And these are the enzymes that catalyze the addition of a palmitoyl residue to a protein. So the question was then, so first of all, what happens in prion infection? And it turns out that prion infection removes the acylation of PIK5. So this is the Acyl-RAC methodology, and what you see is that this is non-infectious brain homogenate. We are looking at acylated PIK5, and what happens is that in the context of a prion infection, the acylation of PIK5 disappears. So PIK5 becomes destabilized because it's no longer acylated. So now the question was, okay, there are 23 acyl transferases. Which one is responsible for PIK5 acylation? And so has been knocked down individually every single mammalian acyl transferase, and he found that there are two that reduce the acylation level of PIKfyve. This is DHSC9 and 21. And they are cooperative because if you knock down both of them, then you really dramatically reduce the acylation of PIKfyve. And it turns out that if you infect the cells with prions, the localization of the acyl transferase is changed completely. So you see here the acyl transferases in the non-infected cells, and here you see that they are completely delocalized after prion infection. So at this point, we are pretty convinced that the mechanism that leads to the vacuolation is really goes through the delocalization of the acyl transferases. And then we mapped the palmitoylation sites, but that's not so interesting. And I will also jump, you know what, I will skip a lot of things here and just mention that if we overexpress the acyl transferases, then we can partially rescue the levels of PIKfyve. So here you see again non-infectious PIKfyve levels. You infect with prions, PIKfyve goes down. You overexpress the DHH9, DHH21, or both, and now actually PIKfyve levels come up. So it's so you can actually restore the homeostasis of PIKfyve by simply overexpressing its acyl transferases in the case of prion infection. So again, what does PIKfyve do? PIKfyve makes phosphorinositol 3,5-diphosphate. So the idea was, okay, if this is really what is needed to suppress the vacuolation, maybe we can just simply deliver the substrate to prion infected cells and suppress vacuoles. So we had a company make for us a body PI conjugate of phosphorinositol 3,5-phosphates. So this is a water-soluble analog and of the which contains the body PI, which is also a nicely fluorescent molecule, and we deliver this to prion infected cells. And the beautiful thing is that look here at vacuolation. These are the GT17 cells. Here you see non-infectious brain homogenate. There are no vacuoles. You infect them with prions, you have a lot of vacuoles. And if you, however, give the BPIP, the body PI phosphorinositol diphosphate conjugate to the prion infected cells, you massively reduce the number of vacuoles. And not only that, what I didn't say is that in prion infection you have a major screw up on the of lysosomal enzymes. You have all these enzymes that are upregulated, so there is a major disturbance of lysosomal metabolism, and this is completely suppressed by addition of the BPIP conjugate, and this is ratio to non-infected cells. So these are prion infected, and these are prion infected with addition of BPIP. And the nice thing, this is, I think, really the most remarkable thing, is that we can actually protect the cerebellar organotypic slice cultures from the deleterious effects of prions by adding BPIP. And this, again, these are the slices, cerebellar slices, infected with non-infectious brain homogenate, you see here, and here infected with prions, you see it's completely destroyed. Here the slice architecture is nicely preserved, but here it's gone. And if you gave BPIP, however, the prion infection no longer destroys the slice, and the granule cells are still alive. So also in the slice cultures, the lysosomal enzymes are rescued. So the bottom line here is really that the final slide, prion mimetic antibodies, which is something I'm super fascinated by, because the antibody is very rapid. The prion infection takes several weeks to destroy the slice. The POM1 destroys the slice in 3 days. You see what happens here. Here we have a slice that was treated with POM1, but POM1 had been pre-incubated with equimolar amounts of prion protein, so it's inactive because its paratope is blocked by the antigen. So POM1 does not destroy the slice. But if you give POM1 without PrP, then you destroy the slice in 3 days. But if you give POM1 to a slice that is being treated with BPIP, with adduct of the reaction catalyzed by PIKfyve, then you actually rescue the granule cells. You see, it's not a complete rescue. I mean, there is still some severe damage ongoing, but the slice is in a much better shape than if you don't give the BPIP. So this is really the model. We think that everything starts with the ER stress, and this leads to deacylation of PIK5 through the delocalization of the acyl transferases. Deacylated PIK5 becomes polyphenylated, and its half-life is decreased. As a result, there is less PIK5 available in the cytosol. This leads to lysosomal dysfunction that activate TFEB, which is a master transcription factors of lysosomal genes. This activates a lot of lysosomal enzymes, which again leads to the alteration of cellular lipids, and this eventually results in the formation of the vacuoles. And this model is validated by the fact that interfering with each one of the steps that I've shown here can prevent formation of vacuoles, either by preventing ER stress with using the GSK compound, or by overexpressing the acyl transferases, or by supplying the substrate or supplying the reaction product of PIK5. Right, I think that I will stop here because I've been talking for an hour already, and wish simply to acknowledge all my co-workers, and it's a big institute. They do a lot of things, including stuff that I've not been talking about, but I really wanted today specifically to tell you about the work of Daniel Kirschenbaum, Francesca Catto, and Ashwin Lakkaraju. And I'm also grateful to our funders, and particularly to the European Research Council, and to the Nomis Foundation, and to the Swiss National Science Foundation, which are supporting the research of my lab. Thank you very much.
Thank you, Professor. Now we can start taking questions. So please unmute yourselves and begin speaking.
I'm going to jump in because I'm always excited, Adriano. So I thank you for this whirlwind tour as per usual. I'm particularly interested by the PIK5 stuff. Do you think it matters what cell type in the brain is involved? I mean, my guess is by the looking of those plots is probably all of them, but do you have anything else to add on that?
Professor, you're on mute. We can't hear you.
Oh, sorry.
There you go.
I pushed some wrong buttons. I hope that my seminar was not muted. This would be a disaster. Okay, no, I think that Shane is a very good point. I mean, is the depletion of PIK5? I think that in any cell, PIK5 is a cell essential molecule, so you cannot knock out PIK5. The cells will die. And in fact, we are doing a beautiful experiment now where we are trying to find mutants. We are doing CRISPR survival screens to find the genes that, if deleted, will permit the survival of PIKfyve knockout. So we are really trying to go deep into the biology of PIKfyve. But at the moment, what I can say is that in any cell, including HeLa cells, and so if you knock out PIKfyve, the cell will die. And yet the vacuoles in Creutzfeldt-Jakob, they are neuronal. They do not appear in other cells. And so so there is a specificity layer there that we do not understand yet.
Hi, we're going to meet later tonight. A wonderful talk. So I just to follow up on what you were just talking about, the spongiform changes are macroscopic. The things that you were showing were, sorry, that's me in the background, are very small.
Yeah.
And then I was looking at the images and I was trying to understand whether are those, so I had a couple questions about how you get from microscopic vacuoles to macroscopic spongiform changes. And is it multiple neurons that are showing those changes to give you something macroscopic, which might indicate that you have juxtacrine signaling between the neurons? And is it the entire neuron, or is it just part of the neuron? So could you speak to that?
Right. So you're right. The vacuoles that we see in Creutzfeldt-Jakob disease are larger. They can be 40 microns in diameter, 50 microns. The vacuoles that we see in the cell culture are much smaller than that. But the markers indicate that these vacuoles are comparable because their surface exhibits all the markers of late endosomes and lysosomes. So I believe that the cellular model, even if the morphology is different, I believe we can make a plausible case that we are talking about a similar phenomenon.
And so those are 40 microns. Do they always contain a membrane around them? That's rather remarkable that something could expand to that extent. Yeah.
Yes. So I would say that electron microscopy evidence from many, many decades indicates, and here many people, Paul Brown, Pavel Libersky, and Manuelidis, many people have actually done electron microscopy since the 1960s about of these organelles. And I would argue, yes, everything speaks in favor of the vacuoles being lined with a lipid membrane, but nowadays there are better ways of looking at it. And what we are starting now is to use STORM, super resolution microscopy, that of course does not have quite the same resolution as electron microscopy, but it has the great advantage that you can look at a lot of different markers. I have to say that for me, this was a bit of a gamble, and it still is. But, you know, vacuolation is such a conspicuous change in all forms of prion diseases that I have been thinking for decades that this cannot just be an epiphenomenon. It is something that has to inform us about the pathogenesis of the disease, and also because it's very specific. I mean, there are not so many diseases that give this pattern of vacuolation. So that was really the primary motivation for looking into it.
Can I ask you just briefly, you mentioned that you're having problems publishing this. I mean, this, having read the preprint, it seems a very strong story that is, you know, supported by all these different approaches that you described to us. What is the main problem that people have with this paper?
Oh, well, you know how it is. I mean, you have editors who wouldn't even send it out for review. So for the time being, I don't even know what the problem is, because nobody has even been interested in even reading the paper.
Now, you sort of you tie this into sort of lysosomal dysfunction. isn't this then seen, these vacuoles, in at least some of the diseases that have been linked to lysosomal dysfunction? Have you sort of started looking into those?
We haven't, but it's a good point, yes. And also I have to say that I have somewhat, for dramatic emphasis, I have said that vacuolation is very characteristic of prions, and I would say that's true. But, you know, in reality, even Alzheimer's disease has something that was recognized a century ago that is called granulovacuolar degeneration. And it's not very common. Not all Alzheimer's patients have it. It's also not as extensive as the vacuolation of the spongiosis of prions. But, you know, it raises the question whether we may be talking about similar mechanisms. So I would love to look at PIKfyve in granulovacuolar degeneration, and then there are other things that I'm super interested. For example, a disease of the muscle, inclusion body myopathy. Inclusion body myopathy is the morphological characteristic is something that is called rimmed vacuoles, and you know, I bet that PIKfyve is involved in that, but I don't know for sure. So that is one of the things that we are going to do next. Yeah.
Yeah,
Adriano, I think you're right about the vacuolation being quite common. Many years ago, we made a conditional mouse targeting the Swiss cheese gene in Drosophila.
Yeah.
It's also called NTE, and we saw huge vacuoles in the CNS. I can show you later.
I want to see that.
Yeah, I was actually going to mention that because Moses published that paper many years ago in PNAS, I think.
Right.
And I was going to ask you about that, but I wanted to briefly, if I can, just comment a little bit on the antibody story. That was quite fascinating. You know, we know that people are asking always, what epitope should you target?
Yeah.
Whether it's A-beta or tau or synuclein or so on. And what we are seeing is that you can have dramatic differences in the efficacy within the same region. You know, basically you're targeting the same epitope, but you can have dramatic differences in, you know, whether you are targeting a certain conformation, I guess, within that epitope. And then you can imagine that when you're doing these tiny sort of modifications, that when you humanize even an antibody, even if you don't change the CTR regions, you do change the conformation of that area, and so you do change the affinity.
Sure.
And then you can imagine that, okay, so you have all these mouse studies that have been done, then people move into clinical trials on the humanized ones that haven't really been tested thoroughly in any particular system except just some affinity studies. And then it's really, you know, really unclear what's going to come out of these trials, that you can't necessarily trace it back to the mouse findings because you're dealing with a completely different entity. And that's kind of, we're actually seeing a little bit of this toxicity now with some of the llama antibodies that we're developing.
Okay.
That when you make an isotypes of them, you can see that for some of them, then you actually make them toxic, whereas the single domain antibody is not. So, but I'm just going to leave that there, and but that was quite fascinating as well.
Yeah, I mean, for me, the most dramatic realization, this was many years ago, was when we discovered that actually the epitope is everything. We had antibodies against the N-terminus of PrP and against this domain in the globular, this pocket in the globular domain. And it turns out that the antibodies against the N-terminus were protective, and those against the pocket in the globular domain were incredibly toxic. So simply by changing the epitope, you would completely change the polarity of the antibody. And this is something that to me was pretty shocking because it has huge consequences also for the design of antibody therapies.